turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to this week's Naked Scientists, the programme where we bring you the latest breakthroughs in science, technology and medicine. I'm Chris Smith. And in the programme this week, the genetically engineered mosquitoes released to fight malaria in Africa, how fake news skews public opinion and who is behind it, and I get to find out what it's like to own an extra thumb.
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
First this week, genetically engineered mosquitoes are taking to the air in an experiment to attempt to curtail malaria in Djibouti. The tiny African nation had all but eliminated malaria just over a decade ago, but rising population and urbanisation has seen disease cases skyrocketing again, making it an ideal venue to test the technology, which uses a genetic trick to kill off selectively the female mosquitoes. This leaves behind just the males, which don't bite humans, of course, and they're unharmed, so they can breed and pass on the trait to other members of the species. I've been speaking with Oxitec's CEO, Gray Franson.
We are the pioneers in using genetics to engineer good mosquitoes, and these are non-biting male mosquitoes. When released, they seek out and mate with wild-type female mosquitoes, and all female progeny, all the female babies of that mating will die. And over a successive number of releases of our good mosquitoes into the environment, we will see a dramatic suppression of those malaria-transmitting mosquitoes. And we're now bringing that technology, that approach to the fight against malaria precisely because all of the tools that we have used to date have begun to lose effectiveness. Mosquitoes have engineered themselves around all of our existing tools or weapons to fight these mosquitoes. And thus we are in a race against time to save lives.
I think they call this a gene drive, don't they? It's one of the ways it's referred to. How do you make that happen?
So in this case, in Africa, there's a spreading malaria-transmitting mosquito called Anopheles stevensii. This particular mosquito is spreading very rapidly in urban settings, in cities where millions of people live. Most malaria cases to date have been transmitted in rural areas, rural Africa, for example. We introduce two genes into the genome. One is a fluorescent marker, just so we can see under a certain microscope where our mosquitoes are. But the magical gene is one that, in essence, allows us to release only males and that kills all female progeny, therefore rapidly reducing the population. It's targeted, it's specific, and has no impact on any other part of the environment or the ecosystem.
How does it kill the females selectively?
The team we have just outside of the Oxford area have pioneered the ability to create, in essence, an on and off switch inside the genome of a particular targeted mosquito. What that on and off switch allows us to do, in essence, manufacture mosquitoes at scale, which requires us to keep females alive so we can create lots of male mosquitoes. And then when we release those mosquitoes outside of our factory or outside of the lab, and in the absence of a certain antidote, the kill switch, in essence, turns on. And the kill switch only turns on and interrupts cell production machinery in female mosquitoes.
And why have you gone for Djibouti to test this? Apart from the fact that levels of this disease are rife there again and they're going up, so it makes it a good target from that point of view. But why specifically Djibouti? There are lots of places where malaria is active.
Indeed. So Djibouti is at the perfect and troubling intersection of climate change, of rapid urbanization, of increased transportation between countries. And when you add all of those things together, it creates a perfect environment for this malaria mosquito that is spreading very rapidly in these urban environments. So Anopheles stevensii has, in essence, invaded Djibouti and has had a deadly impact. They went from near elimination in 2012-13 to now tens and tens and tens of thousands of malaria cases spread by this particular mosquito. Now, in 2018, Djibouti's public health leadership reached out to our team and asked if we would develop a new mosquito, a new Anopheles stevensii Oxitec-friendly mosquito to help them. And that is a request we could not deny. We had to lean into this and help them solve this problem. And frankly, it's not just a Djiboutian problem. This invasive mosquito now has spread across the African continent and continues to enter into densely populated urban environments.
Presumably you've modelled how you expect this to play out as the mosquitoes spread and they carry this kill switch into the females and slowly suppress the population. You must have a sort of trajectory in mind as to what you're expecting to see. So how will you monitor that?
Great question. So the trajectory we hope to see does start in Djibouti. We're basically comparing where we released ours to an area where we didn't release ours, and we will see significant suppression in the area we've treated. Beyond that, we begin scaling up. We demonstrate the effectiveness of the mosquito solution in successively larger environments, and then we begin preparing to scale it for interventions where an Oxitec mosquito plays a direct role in a government's efforts to control this particular vector. Then we begin spreading across the African continent to treat those cities where governments wish to see us deployed.
What's the end point for this? Because there must be a law of vanishing returns applying here where you end up to drive it really down. You have to release so many of these mosquitoes to get to the last vestiges of the mosquitoes that are still breeding. And as soon as you take your foot back off that accelerator, does the whole thing not change direction and come roaring back?
Frankly, for this particular malaria transmitting mosquito, we see this as the primary method by which we will secure control over this mosquito. Now, to your particular question, yes, over time, we will have been successful in knocking down maybe 90% of the population. That last 10% of the population requires fewer and fewer releases of Oxitec mosquitoes because we have fewer and fewer mosquitoes to target. And ultimately, we would hope to create a significant suppression such that we drop that remaining wild type presence below a disease transmission threshold where it becomes either completely unimportant or not a threat to the civilian population.
What about the possibility, given that the mosquitoes have just in our generation adapted and continue to do so, that will drive the evolution of wild type mosquitoes through doing this and will evolve a new generation of anopheline mosquitoes that don't die when they get this genetic switch, and that means we'll end up with a really tooled up mosquito that is going to bypass your blockade?
It's a very good question and one that we are asked often, but the manner in which our technology works, it's a mating-based, species-specific, female-focused solution. We will not see resistance to our particular gene technology. And that's good news. Unlike chemical pesticides or insecticidal properties of a bed net, for example, those are blanket active ingredients or chemicals that are spread time after time after time, giving mosquitoes a chance to generate resistance by way of mutations and normal selection processes. For ours, we are introducing male mosquitoes that will target only the targeted species. And again, all females die, and thus there is no chance for selection on those grounds.
Gray Franson at Oxitec, the company behind the new genetically modified anti-malarial mosquitoes. In recent years, we've all become much more familiar with the threat posed by online misinformation. It can undermine confidence in health initiatives like vaccines and skew public opinion around politics. Now, 2 new papers published in the journal Science shed some light on how potent different sorts of misinformation are at influencing members of the public, and also who is behind their creation. One notable observation to emerge from the research is that information that's not wrong, but could be interpreted the wrong way by audiences, is extremely powerful. To quote the summary written by the journal, factual but misleading vaccine content was 46 times more effective at driving vaccine hesitancy than actually flagged misinformation. Sander van der Linden from the University of Cambridge works on how we're affected by disinformation, and he wrote an opinion piece that was published alongside the new research this week.
So they wanted to see if A, what is the causal effect of being exposed to misleading information about vaccines on people's intention to get vaccinated, and B, how can you estimate the total damage from that? And can we estimate maybe even how many lives could have been saved if people hadn't been exposed to this type of misinformation?
What do we call misinformation, though? Because I put it to you, the Chicago Tribune, during all of this, led with a headline that said a doctor died 2 weeks after having the COVID jab. But it doesn't go on to say, and that was because he was run over or he had a heart attack, which was nothing to do with the vaccine.
Exactly. That headline was one of the most viral headlines because millions and millions of people were exposed to it. And the exact question there is a healthy doctor did die 2 weeks after getting the vaccine, right? But it falsely implies causation where there's only correlation. And that is a well-known logical fallacy. And so that's why it's misleading, even though the words technically are true. But what they wanted to do in this study is find out how persuasive are total falsehoods compared to these more subtly misleading statements. And what they found, this misleading type of headline, true but misleading, was 46 times more damaging in a way than the effect of outright falsehoods on people's intentions to get the vaccine.
Why do you think that is?
Well, from a technical perspective, the answer is reach. So if you look at outright falsehoods, they're not shared by that many people. So their reach is fairly limited. So if you look at the math in their papers, basically what they try to estimate is what's the percent reduction in your intention to get vaccinated, and then they need to multiply that by the actual exposure on social media. And so outright falsehoods have actually quite low exposure, whereas mainstream media that's highly misleading has huge exposure. And so when you combine, the effects from persuasion in the lab with huge numbers of millions of people being exposed, that's where you do quite a lot of damage.
Is the take home then that more responsible reporting is needed? Because it sounds like that was just a shoddy headline that was clickbaity. Is that really what we're talking about here, that we just need more responsible journalism?
Yes, in a way, I think we do need more responsible journalism, and that means, fewer clickbait headlines, fewer emotional manipulation, fewer articles that have a clear or biased slant. But the other thing we need is maybe to broaden our understanding of what is misinformation. And so there's some consensus among experts that it shouldn't be just outright falsehoods, because that's a fairly small proportion of what we see on a daily basis. That the bigger problem is misleading content, right? There's some nugget of truth in there, but it's otherwise manipulative. And I think that is the bigger problem and the bigger part of the definition. So when we think about misinformation, we shouldn't only think of flat earthers, but also about mainstream content that is highly misleading.
And the other paper looks at something very relevant right now, both sides of the Atlantic, that's elections and election bias and spreading of misinformation. They say a staggering amount, 80% plus of the misinformation originated from a tiny fraction.
Yeah, and these are called super spreaders or super sharers. And that's a more general finding that, we find that most of the fake news comes from a small group of highly networked, highly influential individuals supplying up to a quarter of the falsehoods that their followers see in their news feed. So if you're on Twitter, about a quarter of the falsehoods you might see might be coming from a super spreader or a super sharer that you're following, intentionally or on accident. I'm assuming nobody in the audience, your audience is following any super sharers, but if that were the case, that's basically what they found. And now I will say that the difference with the other study is that they focus on outright fake news. So if you put two and two together, what we haven't looked at is who are the super sharers of misleading information, because we know misleading information can be more impactful, but who are the super sharers of generally misleading content? Because they're the ones exacerbating the reach. So you were coming back to the Chicago Tribune headline. That was actually influential amongst anti-vaccination activists. As you predicted, it was a mainstream outlet that published it, but it was pushed by anti-vaccination groups on Facebook. Similarly, there was another study recently that showed that there's about 50 physicians in the US that were responsible for a disproportionate amount of COVID misinformation. So there's these networks of highly influential actors that are amplifying fake as well as misleading content. And in fact, if you look at the motives of super spreaders, they're quite diverse. For some, it's fame. For others, it's financial. And so to what extent we're able to change their motives is an open question. Maybe it's more realistic to try to, somehow intervene to make sure that their followers are not exposed to it. Maybe that's a more, more plausible route than actually trying to change the super spreaders themselves.
So persuading the social network platforms themselves to do something. One person I spoke to, she did a study looking at whether you could add a button, instead of just like and dislike, an I trust this content. And then you're basically pinning your integrity to the message, and people will be a bit more hesitant to pin their integrity on something that might be dubious rather than just like it and share it.
Yeah, exactly. And that's, I think, the finding here that true but misleading content is especially problematic aligns well with the research that we do, which is about helping people spot manipulation in content wherever they may see it. But I was going to say, we ran out of space, so the editors kicked out my final paragraph, which was about this sort of newer study that shows that you can actually try to change the incentives too for super spreaders and their followers by implementing some sort of credibility meter. And so the idea is that, if you keep floating untrustworthy content, that's going to hurt a visible public credibility meter that might be assigned to you. And so nobody wants to put their reputation at stake if it actually means they're going to lose points or they're going to lose likes, or the audience might think that they're not credible. So assigning trustworthiness ratings to social media accounts could provide a different type of incentive, and I think it's not a bad idea.
So Twitter, Facebook, et al. All need a credibility meter by the sound of it. That was Sander van der Linden.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
This is The Naked Scientists with me, Chris Smith. Still to come, I'm going to get my hands on a third thumb. Find out how I fare. Before that though, all life evolves. In other words, it changes in response to selective pressure from the environment. And some examples of evolution are extremely complex because they involve not one but two organisms evolving in tandem, one being a parasite, the other being its host. So what are the rules of engagement in arms races like this? Well, Cambridge University's Rebecca Kilner studies cuckoos. And working with colleagues in Australia, she's found that what began as efforts just to imitate the host bird's eggs has morphed down under into full-blown imitation of appearances and sounds of chicks instead.
Our research focuses on how evolution works. And the way that we do that is to look at pairs of species where one species is exploiting another. So we're trying to understand how one species adapts to exploit another species and how that second species adapts to avoid being exploited. And we're interested in understanding whether there are general rules in how evolution plays out in those circumstances.
And the obvious example must be the cuckoo, because this is a bird that plants its egg in another bird's nest and gets the parent birds to then bring up an offspring, a chick, which is not their own.
That's right, and that's hugely costly for the parent that's looking after the cuckoo chick. They pay the price for doing that. They lose their own offspring. The cuckoo chick kills their own young soon after it hatches by balancing any unhatched eggs or newly hatched chicks in the small of its back and walking up the side of the nest and tipping them out. So the host bird loses its own offspring, and then it pays again because it works really hard to look after an enormous cuckoo chick that it has no genetic stake in at the cost of being able to breed again in the future. What we're interested in finding out is how those arms races play out. Do they always go in the same direction across evolutionary time?
How can you tell that?
Perhaps I could provide some kind of previously on ER context to this. So the study of cuckoos and their hosts has been going on for some time and was initiated in Cambridge by Professor Nick Davies some time ago. And he focused his work on the common cuckoo and its many hosts in the UK. And what he showed was that the arms race is very much focused on the egg stage. So cuckoos lay their egg in the host nest, like a reed warbler. The reed warbler carefully scrutinises its eggs, and if it sees an egg that looks a little bit different from its own egg, then it picks it up and it throws it out. And in that way, it can avoid being exploited by the cuckoo. And that defence has in turn selected for cuckoos that can better hide their eggs in the nest. And so they lay an egg that closely matches the host egg in appearance. Where our work comes in is on a completely different set of cuckoos, which live in Australia. They're called the bronze cuckoos. They're quite small compared to the European cuckoo that we're familiar with. And what we showed there was that there was no equivalent arms race happening at the egg stage. The cuckoo lays an egg that's approximately similar to the host's own egg. But if we were to put a model egg in the nest that looked very different, then the host wouldn't throw it out. And it turned out that what had happened was that the whole thing had shifted on to the chick stage. Unlike the UK cuckoo, the Australian cuckoo had evolved a set of rejection rules that were focused at the chick stage and involved spotting the odd chick in the nest. And they were able to do that by listening to the calls made by the cuckoo chick and also by observing that there was a single chick by itself in the nest, which seldom happened when they were raising their own young. And so this combination of cues would lead the female to dismantle the nest while the chick was still alive and begging in the nest and to build a brand new one. And then the cuckoo chick would slowly starve to death over the next couple of days. And then immediately after it died, the meat ants would come in and dismember the corpse and carry it off to their nest and feed their young with the bits of the body. And meanwhile, the host has started a new nest and a new breeding attempt.
How do you know what the timeline is here? How do you know that it started with egg discrimination? Your egg looks different to my normal eggs, I'm going to chuck it out, versus your chick looks different to my chicks in Australia. Was it that was first and the eggs came second, or do you think they all started with egg discrimination and the Australian version has moved on to chicks? How do you know that's the timeline?
We don't know for certain because we haven't lived for the millions of years over which this arms race has played out, but we can deduce it from a few clues. And the first is that the egg that's laid by the Australian cuckoo looks unlike the eggs laid by its close relatives and much more like the eggs laid by its host. So something has happened at that stage. It may have been that in the past the host rejected eggs, just as we see happening in European cuckoos today. What happens now, we suspect, is that the egg contributes to the decision to abandon the chick. So we did all these egg experiments where we put an odd-looking egg in the nest and observed no immediate reaction from the host. But once we'd discovered chick rejection, we went back to those experiments and we noticed that chick rejection was more likely if we had added an odd-looking egg to the nest for one of our earlier experiments. So what we think is perhaps the hosts remember this weird egg and then they use it as part of their accumulated information about what's going on with their current breeding attempt when there's a chick in the nest, and that informs their decision to reject or to continue to feed the chick.
Rebecca Kilner there, and her paper just came out in the journal Science. Most of us have, at one time or another, wished we had an extra thumb on hand to help with fiddly tasks. And Danny Clode, who designs limb prostheses, thought the same. So she made one. It's basically a rubber robotic thumb that straps onto the bottom of the hand in roughly the opposite position to the native thumb, and it closes up onto the palm or against the little finger. She wanted to know how people would get to grips with using an additional digit, particularly as we move into an era when body augmentations like exosuits that can help us to perform certain tasks or go through rehabilitation are set to become much more commonplace. The result, which was showcased and tested on hundreds of members of the public at the Royal Society's famous Summer Science exhibition, also therefore offers an opportunity to explore neurologically what happens as someone gains a body part and learns to use it. I went to meet Danny and her colleague Lucy Dowdall at the Cambridge MRC Cognition and Brain Sciences unit to test it out for myself.
Cool, so we're going to put a third thumb on you today. I do need you to take your shoes off if that's okay.
Good job I put decent socks on.
Yeah, Cool. So if you take
Not
your
every
shoes
day I turn up to an interview and someone says to get your shoes off.
Yeah, we do things differently here.
I thought this was for my hand. Why have I got to get the shoes off?
It is, but it's foot controlled. So each of your big toes is going to control one, what we call degree of freedom of the third thumb, which is the two kind of movements that it does. So one movement is across the hand and back, and the other movement is kind of towards your fingers and back. And so your left and right toes are going to do that. So this is the device here. So there's a part that's worn on your upper arm, like on your bicep, and then that's connected to a wire that's to the motors that are worn on your wrist. And then there's a hand piece that's kind of got two straps. That one goes between your thumb and index, and then the other kind of around the base of your thumb. And that's going to keep the third thumb on your hand.
Okay, so do you need my sleeves rolled up, or is this jumper friendly?
Yeah.
I mean, I know this is the Naked Scientist, right? But there are limits.
Yeah, we have to be jumper friendly in the UK, don't we?
So this is going over my right arm.
Yeah.
So it's on the forearm at the moment. You're going to put that further up, are you?
Yes, I've got to kind of shimmy it up your arm. Yeah.
Okay.
So then
And
that
then
goes
the hand piece
around
is
your-
going over my fingers.
Yeah, so thumb in. Yeah, that goes on your wrist.
Right, so I now have a strap
thumb in
around my
there.
wrist,
Yeah.
a strap around my hand, and a thing which looks like an exceedingly large thumb, but on the bottom of sticking out from the bottom of my hand. That's sort of, if I had monkey hands, that's the length of the sort of digit I would expect to have.
Yeah, I made a smaller one, but it wasn't quite as functional. So, yeah, it's become bigger over the years.
So I now have, I presume that's a battery pack as well,
It
and now
is,
on the
it's a
back of
battery-
my arm.
It's the battery pack and also the kind of PCB, the electronics that connect to wirelessly to the foot control. And then I'm going to put two sensors underneath your big toes. You've got your left control here. If you press down, there we go.
Oh,
That's
okay,
your left
yep.
control.
So this is, I press my left big toe down, and I'm moving towards the rest of my fingers. With my right toe, I'm moving my new thumb into the palm of my hand.
Yep. Yes. So you can see that the speed is responding quite instantly and wirelessly to the pressure of your big toes.
I actually got it's 3 degrees of movement, isn't it? Because I can go into the palm, I can go outwards in the direction my fingers are pointing if I was sort of pointing my fingers away from my body, but I can also move the device so that it's in any of the positions in those 2 directions. It's not just into the palm or out towards my fingers.
Yeah, I'm kind of I was tricky about that and actually got a bit for free in how it's the thumb is fully flexible, so it's 3D printed out of a flexible material, and so it's kind of you're actively pulling in, and then the flexible material is kind of pulling back out. So it kind of seems like there's a lot more degrees of freedom, but there's only 2 controls and 2 motors. So
Do you reckon it'll hold a microphone? Shall I have a go?
Yeah.
It's quite brave. It's quite
Yeah.
heavy, this microphone. Okay, I'm going to have a
it's
go.
quite strong. You want to go other way around, so you got to change the way that you actually use your hand. So I would go that way. So push
you
your
put-
right toe.
So you're putting the microphone along my hand,
Yes.
so it's going to be grabbed. So let me just try that. So
Right toe. Yeah, and press and hold.
you got to hold
Yeah,
quite tight.
yeah.
But it has got it. I mean, that's quite good. I'm not sure what I would do with the, because it's completely occupying the rest of my hand.
Well,
but
it's a big microphone, but it free. What you're doing is you're freeing up the use of your fingers, especially your pinch grip, for example, which is a super functional part of your hand. The third thumb, can grasp something whilst then freeing up that part of your hand.
So what sorts of use cases would you see for this? I grant you, it's very easy to use this. I'm my first go, I grabbed hold of something, and I feel pretty confident that I can put this thing into any position relative to my hand. That's really easy to do. But what's the use case for this? What sort of applications do you foresee?
Yeah, well, this kind of device is augmentation, so extending the function of the hand. It can kind of grasp an object whilst freeing up your kind of index or rest of your fingers. So, for example, holding a bottle whilst unscrewing the cap, that can be done one-handed. So all these kind of tasks that usually require 2 hands, you can explore just using one hand.
So do you think surgeons might be interested in something like this? Because there are some aspects of surgery where surgeons say, I wish I was an octopus.
I would love to collaborate with surgeons on this, especially, people with professions where they kind of like hyper train with their hands, to then expand the function of a hyper trained hand. Surgeons is an interesting example because they use a lot of assistance, so, there's people that are perhaps being their eyes or other hands during the surgery. And so to augment them and create more fluid function during surgery would be an incredible opportunity.
Lucy, you're the neuroscientist on the team. What's your role? What are you actually doing and trying to find out?
So we're interested in how our experiences and our environment shape our brain. After 5 days of training with the thumb, we see changes in how our hand is represented in the brain. And in our next steps in this research, we're looking at how is the third thumb itself understood within this body representation. And that's really important when thinking about if people are using these technologies in their everyday life, how is that then affecting our biological body?
When you say you see changes in the brain, what are those changes? How does the brain seem to incorporate it into its model of the body?
In our brain, we have a body representation. So, for example, if I move any one of my individual fingers, I'll see an area of my brain devoted to that finger lighting up. And after 5 days of training, we find that after the training, the fingers all become more similar to each other. So we're having a reduced representation of our hand in the brain. But we want to then know, is that because we're using our hand differently every day? Because we know that how we use our hand does affect how it's understood by the brain? Or is it something very specific to the third thumb itself and that being incorporated?
And if I put this on the other hand, which is obviously being controlled by the opposite side of my brain, if I put this on the other hand, does the learning transfer, or do I have to start from scratch with the other side?
So we're actually looking at this right now. We don't have the final data, but we are quite surprised to see it does seem to generalise very well. We've also looked at changing the controller, so moving from the toes to controlling it by the heels. And again, this learning seems to generalise. So there's something about this hand and foot coordination. People are able to learn very quickly and generalise very quickly.
Danny?
With our current research that we're talking about as well, we actually found that we had a really inclusive range of people that tried the third thumb on for the first time at the Royal Society Summer Science Exhibition. And although it was only a right-handed thumb that I had, we included left-handers and right-handers, and everyone had a very similar response to it.
That was why I was asking the question, because I was thinking 90% of the population are right-handed.
Yes.
You've put this on my right hand.
Yes.
Did I find it easy because I am right-handed? If you'd given me a left-handed one, would I have struggled the same way a left-hander might if I gave them a right-handed one, or does it just generalise?
It seems to just generalise, which as the designer of it, I was quite surprised. I'm left-handed, so I use mine on my right hand. I don't know why I chose that, but it was probably so I could work on it at the same time.
Danny Clode and Lucy Dowdall, thanks to them. And that work's just come out in Science Robotics. Over to question of the week now, and Will Tingle has taken charge of this question from listener Michael.
What kills a car battery that hasn't been driven or started in a while? Good question, Michael. Returning to a car after a few weeks or so can lead to a frustrating struggle, as the only thing preventing the car from operating is one flat battery. But why does this happen? We put in a call to University College London's Rod Jervis.
There's a process called self-discharge, which all batteries undergo to some extent. And then there is sort of the battery becoming flat, fully degraded and not possible to be regenerated. And that can unfortunately happen with lead-acid batteries, which are the type of sort of starter batteries in internal combustion cars. So the process of self-discharge is really one of fighting against the nature of all batteries. When we charge batteries up, what we're doing is putting them into a semi-unstable state, thermodynamically speaking. And this is the equivalent of pumping water up to a hill, up to a high reservoir. Okay, we're giving the battery that potential energy, but it also means it's in a state where it really wants to get rid of some of that energy. And in some cases, that can lead to favoring some what we call side reactions that end up using a small percentage of the charge stored in a battery. So that happens in lead-acid batteries, lithium-ion batteries, lots of different types of batteries. Some of them lose their charge relatively quickly and have a shelf life of maybe a few months. Others will lose just a couple of percent in a year or something like that. Most often it's reactions with the electrolyte. This is the sort of liquid in the batteries that carries the charged ions back and forth between the electrodes. And then often that will use a very small percentage of the capacity. These side reactions, these self-discharge reactions, are more likely to happen when the battery is in a high state of charge. And so leaving the battery very, very charged will accelerate those reactions, as will increase temperature. So what we can do to avoid that is to maybe store the battery partially charged, not leave it in high states of charge for a long time, not overcharge it, not leave it in warm conditions, etc.
Sage advice there. Thanks to Michael for the question and Rod for the answer. Next time, we're answering this question sent in by listener John, who asks, why is it that the body replaces its cells every year or so? It's not clear why this is beneficial from an evolutionary perspective. My old arm was working just fine, thank you very much. A valid observation, John, and we shall have an answer for you next time.
Thanks very much to Will Tingle. And if you think you know the answer or you have a question of your own, then why not drop us a line? It's chris@thenakedscientist.com. You can also pop into our forum at nakedscientist.com/forum, where this and questions like it are being discussed right now. And that's where we have to leave it for this week. Do be sure to tune in on Tuesday, though, when we're going to look at our latest attempts to map the matter and energy of the dark universe. Before I leave you, though, please do, if you can, consider making a donation to us if you like this programme and like the work that we do and would like to support us. We've made it very easy, safe and secure. You just go to nakedscientist.com/donate. And while I'm saying this, I'd like to pass on our extreme thanks to those people who continue to support us month after month. It really means a lot. Thank you. The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. We're also supported by Rolls-Royce. Thank you very much for listening, and until next time, goodbye.